Hello everyone. I'm starting with the first topic that is generally taught in BDS, the first chapter, development of tooth. So I'll be starting with a brief introduction. What happens is that you have a dental lamina. This is a structure in your mouth and the fetus. This later gives rise to bud-like organs, and they transform from bird-like shape to cap-like shape, and this is where the tooth grows. So this would be the area where the tooth is formed. This is a very rough outline of what happens in development of tooth. Now moving ahead with the actual chapter. So this is the introduction. What this Orban textbook starts off with. So they show you a stomodium, which is a primitive oral cavity. What means that it's the oral cavity of fetus. Okay, so this is the oral cavity of a fetus, and it has an outer ectodermal lining. Okay, which is below which. So over here below which the blue color thing, there is connective tissue and connective tissue, which is ectomesenchymal in origin. It is derived from neural crest cells. Okay, neural crest cells are cells which are formed during neurulation. We'll study that in embryology. So these cells, this blue thing, this is the neural crest cells. Derived from the neural crest cell, it's called ectomesenchymal tissue. Okay, then going over here, this is the buccopharyngeal membrane. It connects the oral cavity and the foregut. Okay, and it ruptures in the 27th day of gestation. Then the connection between the oral cavity and the foregut is established. The foregut is lined by endoderm. So this yellow thing, it is the endoderm which lines the foregut. Another important thing is that this connective tissue, ectomesenchymal in origin, this instructs the oral cavity or the dental lamina when to form the tooth and where to form it. Okay, and the tooth development, so it begins in the anterior portion first. Obviously, this firstly the central incisors and canines are formed first. It's just that. Now coming on to the dental lamina. Okay, what is the dental lamina? When the buccopharyngeal membrane ruptures on the 27th day of gestation, so after 3 weeks of the rupture, what happens is that you see these outgrowths of the oral ectoderm. So if I just zoom out, you saw here this was like there was a pinkish structure over here. Okay, this is the oral ectoderm and this is the ectomesenchymal cells. So I have just zoomed this out over here. This is the oral ectoderm and this is the mesenchymal cells, ectomesenchymal cells. Okay, so you see these proliferations, these outward growths of the oral ectoderm into the ectomesenchymal cells. And how this happens is, so you have your mouth, which is entirely oral ectoderm, and then you will see these growths. Okay, you will see these growths happening in this. Sorry, this horseshoe shape. These happen in the horseshoe arc shape like this. Okay, so this forms, you can say it forms a band kind of. So this band, this is called primary epithelial band. So this is the shape of your arch. Okay, the maxilla and the mandible. So these outgrowths take place in the shape of that arch. Okay, so this is called primary epithelial band. Now, in the seventh week, so I have shown this the dental lamina, which is present in this horseshoe area. Okay. Now, in the seventh week, what happens is that this outer portion of the dental lamina, it proliferates into the vestibular lamina. Okay, this differentiates into something that is called the vestibular lamina, and the inner, the lingual portion proliferates into the dental lamina. Okay, now what happens is that after this differentiation, even the dental lamina, so if this is the dental lamina, this is the dental lamina, I'll just erase this so you can see that the dental lamina was blue and in the diagram, of course. So the dental lamina, it has two further differentiations. Okay, so it has a distal extension and it has a lingual extension. What does the distal extension do? So the distal extension gives rise to the permanent molars, and the lingual extension, the permanent tooth other than the molars are formed. Okay, and then what happens in the dental lamina? They in there are buds of the deciduous tooth, only the deciduous tooth. Okay, so you would see I have made this diagram. What happens is, so this is the dental lamina, this is the lingual extension called the successional lamina, and this is the distal extension. Okay. Now what happens is that this is the tooth bud of a deciduous tooth. So from here a deciduous tooth will arise, and this tiny circle over here, it is the bud of a permanent tooth. Okay, so it just lies opposite to the bud of the deciduous tooth. uh, and then there is the vestibular lamina. It will hollow out. It's also called the lip furrow band, so that the space between your gums and the cheeks, vestibular lamina forms that. Okay. Now moving on. Uh, some details about the successional lamina. It develops from fifth month in utero to 10th month of age. Okay, so successional lamina gives rise to all the permanent tooth. So the permanent tooth, the central incisor, starts developing from fifth month in utero, and the second premolar, which is premolar, it develops to 10th month of age, and the molars which are developed later on or at different time, like the third molar is developed later on, so it arises from the distal extension. All right. And now coming on to the fate of the dental lamina. What happens is the activity of the dental lamina is about 5 years, because the last teeth that is formed, that is the third molar, it starts development around 4 to 5 years. Okay. But what happens is that if the central incisor has been developed, so the activity over there is not required. So the dental lamina over there, it will degenerate. Okay, it won't be there. It won't be active in the central incisor region, but it will be active in the distal extension where the molar is developing. Okay. And the last thing, that the remnants of dental lamina may persist in the form of pearls, which just means like tiny spherical ball kind of structure, and they are called cell rests of Ceres. Okay, and they exist in the jaw and the gingiva where dental lamina exists. Okay, now coming on to the vestibular lamina, I just told you that it hollows out and becomes the space between and join the lip. Okay. Now coming on to tooth development. What happens? What forms the tooth? We are going to discuss that. Okay. So here is your dental lamina, and this is the bud-like structure which gives rise to the tooth. Okay. This bud-like structure is called the enamel organ. Okay. Enamel grown organ is the downgrowth from the dental lamina into the ectomesenchymal region. Okay. It starts first in the anterior region. I have told this before. What this enamel organ later on forms, it's the tooth germ. Okay, this is a tooth germ. This is the main structure. What constitutes a tooth germ? So a tooth germ consists of enamel organ, dental papilla, and dental follicle. So what is dental papilla and dental follicle? This region, this condensation of ectomesenchymal area is dental papilla, and this is this area around the enamel organ. So this yellow thing is the enamel organ. Okay, and the thing around this is called the dental sac or dental follicle. So you might be thinking that what is dental papilla? It's just cells. So it is just cells. It's just that so this was in a bud-like thing. Okay, then the enamel organ, it grows into a cap-like structure, and this ectomesenchymal cells that stay in here in this hall, in this area, in this cap-like area. Okay, so they are very dense. They have a lot of fibers and other things. So they are very dense, and this becomes the dental papilla. This is called the dental papilla, and the area surrounding this whole enamel organ thing, this becomes the dental sac. Okay, and now, one important thing, that the enamel organ, it is ectodermal in origin. How is this ectodermal? Because we saw that this bud-like thing developed from this oral ectoderm, and the oral ectoderm was derived from the ectoderm. It's called the oral ectoderm. Okay, so it was derived from the ectoderm, but the dental papilla, this is the ectomesenchymal condensation. Okay, this is what existed over here, this blue thing, and this blue thing came from this neural crest cells, which was ectomesenchymal in origin. Okay, so the dental papilla and the dental sac, they are ectomesenchymal in origin. All right, so this is a very important thing, that the tooth germ, it is ectodermal and ectomesenchymal in origin. Now going to dental lamina, sorry, enamel organ. So the enamel organ gives rise to the enamel, okay, and the dental sac, it forms cementum, periodontal ligament, and alveolar bone. And the dental papilla will give rise to dentine and pulp. You have to remember this. And if someone asks like, is cementum ectodermal in origin? So that would be false, because the cementum would be ectomesenchymal in origin, because it originated from the dental sac. All right. Now this thing. So this is the a tiny tissue. This is dental lamina. This comes from dental lamina, which attaches this tooth germ from the oral cavity and the oral ectoderm. What happens is when the this enamel organ, not the not the enamel organ, but when the tooth germ is completely ready, it is in a bell shape. You will see later on in the structure, the development of tooth. So it will be in a bell kind of shape. You see how a tooth is developed. So it will be in this kind of shape, kind of. You can see like this would be a central incisor over here, and this would be the shape of the tooth gem. So when a tooth gem is developed up to that stage, what happens is that this dental lamina, it will thin off and it will degenerate. So entire contact of the tooth germ from the oral cavity will be broken off. Okay, it will be on its own. So the tooth germ loses its connection with the epithelium of the primitive oral cavity, and development of tooth results from interaction of epithelium derived from the first arch and the ectomesenchymal cells derived from the neural crest cells. So this is a line in the organs. What it means is that, um, so you might be thinking, okay, so a tooth develops from interaction between the epithelium derived from first arch and the ectomesenchymal cells derived from the neural crystals. So we saw that there was the oral ectoderm, and then there was the ectomesenchymal cells. So it arises from the interaction of these two layers. Okay, these two layers. But what happens is that they're telling you basically that this is derived from cells of a first arch, and this is derived from the neural crystals. All right. And what is the first arch? So when an embryo is developing, you will study this in embryology. So when an embryo is developing, so let's say this is the face of the fetus, and then this is the body. Very rough diagram. So it will have some foldings over here. Okay, it will have like this folding one, two, three, four. It has these foldings, and these foldings are called arches. So these arches give rise to almost every structure in your body. So this is the first arch. This is the second arch. The second arch will, let's say, give rise to heart. So the first arch will give rise to this oral epithelium. We're telling you this the origin of this oral ectoderm. Okay. So this is it for the first topic, and I'll start with the second topic. Hi everyone. So today we'll be starting with the stages of tooth development. Today I'll probably start with bud stage and go on up to early bell stage, and I'll cover advanced bell stage and root formation in another video. So just giving you a brief insight. Tooth development takes place in four stages majorly. First is the bud stage, second is the cap stage, third is the early bell stage, and fourth is the advanced bell stage. What happens is that after the dental lamina starts proliferating, it forms these ovoid swellings into the ectomesenchyme, and it takes kind of this bird form, and then it takes this cap form. So these stages are named upon the morphology of the tooth germ in that particular stage. Okay, these are called the morphological stages of tooth development, and there's something called physiological stages of tooth development that I'll study later on. Okay, now moving on. There is this line in the organs which says while the size and shape of the individual teeth are different, they pass through similar stages of development. So as we know, the incisors are smaller and the molars are bigger, but they all have similar stages of development. Okay. Now moving on, the first stage, which is the bud stage. So the bud stage starts with the differentiation of dental lamina. So this is the dental lamina, and then there's a basement membrane, and there is ectomesenchyme. So because of the interaction of the dental lamina and ectomesenchyme, the dental lamina proliferates into the ectomesenchyme, and it proliferates into 10 places throughout our arches. Okay, so we're talking about deciduous teeth formation. Okay, we have 10 deciduous teeth in each of the arch, so it will proliferate along 10 points. Each proliferation or each bud represents a deciduous tooth. So if we zoom into this bud, what we'll see is that there is the periphery is lined by low columnar cells, and the center is filled by polygonal cells. All right, and then this is the ectomesenchyme, so it shows some condensation in this stage. Okay, so the same information is written over here, that it has low columnar cells and polygonal cells, and because in the ectomesenchyme there is mitosis, migration of neural crest cells, and condensation of ectomesenchyme, which will form the dental papilla and the dental sac. Okay, moving on from the bud stage, and the tooth enters the cap stage. Okay, what happens in the cap stage is that, so imagine this was your bud, there is unequal growth of the bud. Okay, it will grow from these two sides and not from over here, which forms due to the unequal growth. There is a shallow invagination. This is the shallow invagination which is formed in the cap stage. Okay, so this is the statement written that unequal growth in different parts of the tooth bud leads to the cap stage, which is characterized by a shallow invagination in the deep surface of the bud. Okay, what other features are present in the cap stage? So we had low columnar cells over here. Now this is converted into outer enamel epithelium and inner enamel epithelium. So what are outer enamel epithelium? They are cuboidal cells in here, and inner enamel epithelium, they are tall columnar, and these cells, these will form ameloblast. Okay, these will later differentiate into ameloblast, and the ameloblast will form enamel. So these are enamel forming cells. These are primitive form of enamel forming cells. Okay, what else is here? So this is the stellate reticulum. So we had some polygonal cells in here. These are converted into stellate reticulum. What is stellate reticulum? These are star-shaped cells, and how are they formed? So the polygonal cells, they absorb a lot of water. And why do they absorb a lot of water? Because we have a lot of glycosamines in the matrix or the ground substance in here. So when they absorb a lot of water, they are converted into star-shaped cells. And this entire network, they are all connected to each other through desmosomes. Okay. So this entire network of the star-shaped cells, this is called stellate reticulum. What is the function of stellate reticulum? So they have a cushion-like consistency. Obviously, they are filled with water. So they act as a shock absorber, and they protect the enamel-forming cells. These inner enamel epithelium. This is the enamel-forming cells. So it protects them. Okay. What else is happening in outside the enamel organ? So this is the dental papilla. Okay. It will show. It was showing condensation in the previous stage. Now there is increased mitotic activity. Capillaries start developing, and some cells peripheral to enamel organs enlarge. Okay, what will happen is that you see this darkened area. Okay, the cells in here, they are going to be converted into odontoblasts. Okay, these cells, what will happen is that this inner enamel epithelium, it will when it's converted into ameloblast, it will send signals into this area, and these cells will be converted into odontoblasts, the dentine forming cells. So before this happens, these cells in this area, the primitive odontoblasts, they start enlarging and differentiating. Not differentiating, but they start enlarging. and there is capillary budding so that they can get nutrition. Now, and this is about the dental papilla. What is happening in the dental sac? What's happening in the dental sac? So, what was the dental sac? It hasn't been formed yet. So, the marginal condensation of ectomesenchyme starts. Okay, and there's more fibers and layers start to develop, and this a primitive dental sac capsular structure kind of starts developing in the cap stage. This information it's just regarding the polygonal cells that how they are converted into and the stellate reticulum. So you can read that. Now moving on to the transient structures. Okay, what are the transient structures? Uh, Transient structures are seen in the cap stage and they disappear before the enamel formation begins. So in the next stage, enamel formation and dentine formation will begin. So these structures are seen only in the cap stage and they disappear. What are they? So there is the central cells of the enamel organ. Okay, cells in the central of enamel organ, they're very densely packed and they are called enamel knot. Sometimes they project outside the enamel organ into the dental papilla and they show a knob-like structure. They may act as a reservoir of cells, so they have a lot of cells and this is continuously growing. So this can provide cells to the entire enamel organ, and they also act as a signaling center and play an important role in determining the shape of the tooth. Okay, now moving on. So this will have a vertical extension. So this I was just talking about this part. This was the enamel knot. Now this is the enamel cord. This is the vertical extension of the enamel knot. And when this extension reaches the outer enamel epithelium, it is called the enamel septum. Because at this point it has divided the entire enamel organ into two parts. So this acts as a septum and this is called an enamel septum. Now upon the point of meeting outer enamel epithelium and this enamel septum, a depression is formed. This depression is called enamel navel. So these are the transient structures and they disappear before enamel formation. Now the bell stage. What happens in the bell stage? So this is obviously the most significant stage, and here I'll be talking about the early bell stage. What happens is the in the bell stage that crown shape is determined. So until now, all the tooths they had like a bud stage, then they all had a cap stage, and they all looked similar. But now, if I want an incisor, okay, so the incisor for the incisor, the bell stage will kind of look like this. Okay, it will look kind of like this. If I have a molar, then it will look kind of like this. So in this stage, the shape of the tooth is determined. Okay, now how is it determined? It is determined because of the genes and their signaling molecules. So they determine which teeth is formed and where it is going to be formed. Different crown shapes is due to differential rate of mitosis and okay. So if I have a different shape, obviously this will the enamel organ will show more growth over here. So the shape is due to the different rates of mitosis and differences in differentiation. Cells begin to differentiate only when the cells cease to divide. So what will happen is first the enamel organ will grow, okay, and then it will start forming the enamel, and then the dentine will be formed. So what it's saying that before the enamel and the dentine and before the ameloblast and the odontoblast are formed, first the entire growth will take place and then the differentiation of ameloblast and odontoblast will take place. Okay. The inner enamel epithelium cells which lie in the future cusp tip or incisor region stops dividing early and begin to differentiate first. So the cells in here, and you say this is the area with the incisor tip. Okay, the cells in here they will start dividing. They will start differentiating first. And this area may still start to keep growing, but these will start differentiating first. Now this diagram is a very complex diagram, but it has all the information regarding the early bell stage. So you can write down your answers in these headings which I have mentioned in the form of diagram, but you can write your answers in a paragraph form and you can mention all of these as headings. So starting with what's new, we had this kind of structure up till cap stage as well. What is this? This is successional lamina. and this is new in the early bell stage. So what does it represent? It represents the bud stage of the permanent successor of the teeth. Okay, so imagine if this is a central incisor, this is a germ of central incisor, of a deciduous central incisor. So sometime it will be replaced by a permanent central incisor. So it is the bud of the permanent central incisor that will replace this. Okay, this is called successional lamina and it is visible in early bell stage. Now there is this new layer called stratum intermedium. Okay, this layer, it is a multi-layered structure. It is formed over the inner enamel epithelium, and what does it do? So it has a very high degree of metabolic activity, and it is essential for enamel formation. So that is the main function. It is essential for enamel formation, and it is absent in the part where enamel is not formed. That is the root. So it is absent in the root portion. Now, in the enamel epithelium, um, what changes are shown in this stage? So it is single layer of cell. It will differentiate into tall columnar cells before amelogenesis. Okay, so before amelogenesis, it will form tall columnar cells called ameloblast, and these are 4 to 5 micrometer in diameter and 40 micrometer in height, attached to one another through junctional complexes. So these red dots, these are attachments called junctional complexes, and they're also attached to the stratum intermedium through desmosomes. The desmosomes are also attached to each other through, the stratum intermedium cells are also attached to each other through desmosomes. Okay, they exert an organizing influence on the mesenchymal cells, which later become odontoblast. So I'll explain this. What will happen is that these inner enamel epithelium cells, okay, they will form ameloblast, and let's just say that this has formed. Now this is an ameloblast. What this ameloblast will do? It will lay an organizing influence over this dental papilla. And what will the organizing influence do? It will cause the cell of the dental papilla to differentiate into an odontoblast. Okay, so now this is an odontoblast, and this odontoblast will then lay dentine. So this will form dentine in here, and this dentine will then influence the inner enamel epithelium to form enamel in here. Okay, so this is how the whole process goes. So in that description, what's written is that basically these ameloblast will influence the dental papilla cells to form odontoblast. Now moving on, cervical loop. This is a new structure. So this is just the junction where the inner enamel epithelium meets the outer enamel epithelium. It is an area of very high intensity mitotic activity, and this will play a very important role in root formation later on. Okay, so this is where the heart with roots epithelial sheet will be formed. We'll see that later on. Now, stellate reticulum, it undergoes some major changes. What happens is that it expands further. Uh, it expands further due to more water entering the stellate reticulum. Okay, but then it collapses as well. So it collapses, and what happens upon it collapsing is that this outer enamel epithelium and the inner enamel epithelium, the distance between them decreases, and it has to decrease because, as I explained to you, that dentine will be formed in there, right? So until now, the enamel organ was getting supplied through the dental papilla, all the nutrient supply. But when once the dentine will be formed, the nutrition supply will stop. So the enamel organ will try, and the inner enamel epithelium will try to derive the nutrition from outside. So to derive the nutrition from outside, this stratum intermedium will collapse and they'll come close together. So it's easier for the inner enamel epithelium to draw the nutrition from outside. Okay, and so it expands further, collapses, and reduces the distance. Okay. Uh-huh. And then what happens is that the collapsing happens first near the incisor edge and then it will continue towards the cervical edge. Okay. Now going on, outer enamel epithelium. So now in this stage they are flattened to a low cuboidal form. Okay, and as I explained that it will be collapsing and this will come together. So when this will come together, it will form, they'll come closer, the outer enamel epithelium and the inner enamel epithelium. So it will be in folds. Okay, so this won't be such a smooth structure. It will become like a kind of like folding. There will be foldings in the outer enamel epithelium, and the dental sac forms papillae that contain capillary loops and provide rich nutrition supply to the avascular enamel organ. So I'll just draw a tiny diagram that what will happen to the enamel organ. So it will, so the sterate reticulum has collapsed. Okay, the outer enamel epithelium now it has come closer to the inner enamel epithelium, and the dental sac. So this portion is the dental sac. Okay, it is outside. It will kind of try to get in here. It will form papillae, and it has a lot of capillary supply. So the capillary will try to supply the inner enamel epithelium and the stratum intermedium. Okay, so this is what will happen. Now, moving to the next slide. Dental papilla. Okay, so some changes will happen in the dental papilla and the dental sac as well. What will happen in the dental papilla, as I already explained, that the inner enamel epithelium. So let's say it has organized, it is converted into ameloblast now. So uh, it will send off signals to the dental papilla, and this will start differentiating into odontoblast. So the first form of the odontoblast, it's a cuboidal form. So these cells will first take a cuboidal form, then they will form a tall columnar form. Okay, and these tall columnar, when they have the potential to secrete dentine, they are called odontoblasts. So this is how differentiation of the odontoblast will take place. There is a basement membrane separating the enamel organ and the dental papilla, and just prior to dentine formation, this is called the membrana preformativa. Now the dental sac. So before dental tissue formation, dental sac shows a circular arrangement of its fibers and resembles a capsular structure. So in the cap stage, we saw that it was showing condensation of the fibers. So dental sac, it basically forms a capsular structure around the enamel organ and a very dense structure of fibers around it. Okay, with root development, these differentiate into periodontal fibers that become embedded in the cementum and the alveolar bone. So these fibers, so right now the crown is developing. When the roots start developing, these fibers will form periodontal ligament, and they will connect. They will be embedded in the cementum and the alveolar bone. Okay. So I will cover. Hi everyone. So today I'll be starting with the advanced bell stage, and this will be the last video for the chapter of development of tooth. So I'll just explain to you what happens in this stage first. So in the previous slides we saw that there was a membrane between the dental papilla and the enamel organ. This membrane was called membrana preformativa. This membrane disappears in the advanced bell stage, and what happens is we have this internal enamel epithelium which is already converted into ameloblast by now. Okay, this ameloblast induces the dental papilla to form odontoblast. Okay, so now these odontoblasts are formed. What will these odontoblasts do? They will lay dentine, pre-dentine. The first layer of dentine is laid. Now this dentine induces the ameloblast to form enamel. Okay, so this layer of enamel will be formed in here. So what happens in advanced bell stage? A beginning of enamel and dentine formation starts in the advanced bell stage. Now membrana preformativa disappears. Future dentino-enamel junction is determined because it is the boundary between the dentine and enamel. So this is determined in this stage. Okay, uh, and formation of dentine takes place in first in the cuspal region and then moves pulpally and apically. So first the dentine will be formed in here, around here, and then it will move pulpally and apically. So it will move towards this direction, the formation of dentine. Okay, and the internal enamel epithelium has already differentiated into ameloblast, and the first layer of dentine is laid. So I already explained to you the process. This is the whole format of that. Enamel is laid coronally and cervically. So enamel is first laid in here. Okay, it is laid in downwards direction and in this direction. And this is the direction of enamel formation. And development of root begins after the formation of dentine reaches the cemento-enamel junction. So root will only be formed after this entire area has been filled with dentine and this has been filled with enamel, the outer area, and when the formation of dentine reaches the cemento-enamel junction, then the formation of root starts taking place. Now moving to the Hertwig's epithelial root sheet. Okay, so this is how the formation of root will start taking place through this structure. What is the structure? What happens is, so now your enamel is formed. Okay, sorry, your dentine is formed. This is the dentine and this is the enamel. Now this is formed and it has reached this cemento enamel junction, as I said in the previous point. So now formation of root will start taking place. So what will happen that this enamel organ, we had the in the enamel organ we had this, these cells were there, no? The outer enamel epithelium and the inner enamel epithelium. So this tiny part, I'll explain in here. So this tiny part in here, it has inner enamel epithelium and outer enamel epithelium, and this part bends. Okay, it forms a 90-degree angle, and this is called Hertwig's epithelial root sheet. Okay, so this is the Hertwig epithelial root sheet, and this will form the entire root. So I will explain to you how first the root is formed and then I'll explain to you the HERS. So this is the structure of the Hertwig's epithelial root sheet. It forms like a 90-degree bending or in the enamel organ. Okay, so it's written the cervical portion of the enamel organ gives rise to the HERS. So this is the cervical portion and the layer bends in here. That is the formation of HERS. Before root formation, the root sheath forms epithelial diaphragm. So this bending, this portion is called the diaphragm and it is formed in here before root formation. The inner enamel epithelium induces the dental papilla to differentiate into odontoblast. Okay, so what is happening over here? Okay, so the dentine and enamel is formed in the coronal portion, in the crown portion. Now this diaphragm is formed. Okay, this bending is formed in here. Now this bending will grow downwards. So this is my enamel organ, and this bending was present over here, but now it will grow downwards. And as it grows downwards, so if I zoom in over here, this was the bending, this was the in inner enamel epithelium, this was the outer enamel epithelium. Now what will the inner enamel epithelium do? Since this area is also dental papilla, okay, so it will induce odontoblast formation, and now when it will induce odontoblast formation, dentine will be formed. So this is what's written in here, that the inner enamel epithelium induces dental papilla to differentiate into odontoblast. Okay, lengthening of the root sheath along with formation of odontoblast take place. So this root sheet keeps growing downwards. Okay, and the dentine formation is happening simultaneously. Odontoblast lay dentine which causes disintegration of HERS. So as soon as dentine is formed in here, this actually causes a breakdown of the HERS. So over here you see the dentine is formed and the HERS is broken down. When the breakdown happens, what happens is that the outer connective tissue, the connective tissue that was outside the dental papilla, this will come in contact with the dentine, and when this comes into the contact with dentine, this differentiates into the this differentiates into cementoblast. So this was tissue of the dental sac. Okay, and now since it came in contact with the dentine, this area will differentiate into cementoblast and this will lay cementum. I'll show you over here. So the dentine came in contact with this tissue, and the tissue, it started forming cementoblast, and those cementoblasts, they started laying cementum in here, and now cementum is formed outside. So this is the whole process how root is formed. All right, just a second. Okay, So due to the disintegration of HERS, the connective tissue of the surrounding dental sac comes in contact with dentine. This causes differentiation of the connective tissue cells into cementoblast and cementum is laid. So I explained to you the formation of enamel, dentine, and now cementum. This is basically how the entire tooth is formed. Now, some details about the HERS. It comes in as a short answer, short note as well. So HERS is the Hertwig's epithelial root sheath. It is two-layered structure derived from most apical part of the enamel organ, which plays a significant role in root formation. So you saw that it had a two-layer and it forms the root. Okay, functions of HERS. It outlines the future root and determines the shape, size, and number of roots. Okay, so it outlines. Obviously, it keeps growing downwards, and that's how the root is formed. It will determine the shape, size, and number. I'll explain to you how it will determine the number in just after this. HERS is formed from enamel organ. It molds the shape of the root and initiates radicular dentine formation. So I showed that to you as well. HERS consists of outer enamel epithelium, inner enamel epithelium. The inner enamel epithelium remains short and does not produce enamel. Okay, so the inner enamel epithelium was not converted into ameloblast and it did not lay enamel in this scenario. It did that in the crown portion but not in the root portion. This induces difference. It only induces differentiation of dental papilla into odontoblast to form radicular dentine. Okay. Now, fate of HERS. What happens to HERS? So, as the first layer of dentine is laid, HERS breaks down, and its remnants may be seen in the periodontal ligament of unerupted tooth and known as rest of Malleus. So as I explained over here that the HERS was broken down, okay. So some remnants of this broken down pieces may remain in the periodontal ligament. So this portion will be later attached to periodontal ligaments, okay. So this may remain and it's called the rest of Malleus. What is the another scenario? So sometimes it may stay adherent to the dentine surface and differentiate into fully functioning ameloblast producing enamel. This is known as enamel pulse. So right now I explained right that this inner enamel epithelium does not convert into ameloblast and it does not lay down enamel. But sometimes it may happen. The HERS was broken down immediately after dentine formation so that the dentine does not induce the formation of ameloblast, the formation of enamel. Sorry, but but if it stays stuck to the dentine, it will be converted into ameloblast and then enamel will be formed. And in such situation, the enamel is called enamel pearl. It won't be formed throughout the root, but it will be formed in some portions. Okay. If the HERS is not properly formed or is broken down before the dentine formation, a defect called accessory root canals on the periodontal surface of the tooth arises. Okay, so in the first scenario, the remnants remained. In the second scenario, it stayed stuck to the dentine. And in the third scenario, it was broken down before the dentine formation. So if it's broken down before the dentine formation, root canals will be formed. Okay, it's just a gap. The dentine will not be properly formed, and root canals will be formed. So these are the three fates of HERS. Now, how does multi-rooted tooth formation take place? Okay, we saw that there is one diaphragm forming and one single root will be formed. But how will more than one root, how will two or three roots will be formed? So what happens is that we had this HERS, okay, this extension. This is how it will look if I see from here. If I see from here, so this entire thing will look somewhat like this. What happens is that in this thing, two extra tongue-like projections are formed. If I want to form more than one root, so if I want to form two roots, these tongue-like projections will be formed and they will eventually meet. And then there will be two apical foramen. So this whole, this is the future of apical foramen, right? So then two epitheliums will be formed and they will fuse together and they will give rise to double rooted tooth, two rooted tooths. And if I want to form three roots, so what will happen? Three such diaphragms will be formed and they will be fused in the center, and then three roots will be formed. One, two, three. Okay, this is the entire process. How do I write it? So in multi-rooted teeth, differential growth of epithelial diaphragm takes place. So differential growth means that there is some extra specialization over here. Some tongue-like extensions of the horizontal diaphragm develop in the case of two in the case of lower molars and three in the case of upper molars, because the lower molars have two roots and the upper molars have three roots. So there will be two diaphragms in here and three in here. These extensions then grow towards each other and fuse and the single cervical opening is converted into three openings. So they fuse together and then this one hole is converted into two holes or three holes. Okay. Towards the pulpal surface, the dentine formation starts over these extensions. So now this is formed and you might say, okay, this is formed in here, but how does dentine formation start? So when this fusion occurs, then dentine formation starts taking place in this direction over these extensions. Okay, so these extensions will give rise to dentine in here, and over here. Now, physiological stages of tooth development. So this is the last topic for this chapter, and this mainly comes in as a short note. So what we studied was morphological stages of tooth development. So it was based on what the enamel organ looked like. But now we will talk about what was happening inside the cells. So that is physiological stages of tooth development. So the first stage initiate, and we'll correlate it with the morphological stage just to understand it better. Okay. So the first stage was initiation. This stage marks the beginning of tooth development initiated by the interaction of oral epithelium and underlying mesenchyme leading to the formation of dental lamina. So physiological stages is mainly based upon the chemical interactions or whatever like the tiny signalings that is happening between the cells, that is the physiological stages. So in the first stage there is just interaction between the oral ectoderm and the ectomesenchyme. Okay, now the second stage is proliferation. During this stage, the dental lamina grows into buds and rapid proliferation of the epithelium into the mesenchyme. So we saw how we had an I'll just draw the diagram how we had oral ectoderm, and then we had the mesenchyme, and then this ectoderm like proliferated into the mesenchyme. So this is the proliferation stage. Okay, now the histodifferentiation stage. In this stage, the cells within the dental bud begin to differentiate into specific cells such as ameloblast and odontoblast. So the whole thing, how we had the whole bud stage, cap stage, early bell stage, and advanced stage. The whole three stages where the entire formation was taking place. So this is the histodifferentiation. Okay, and morphodifferentiation, here the shape of the tooth begins to take form as cells continue to differentiate into different structures including enamel, pulp and cementum. So right now there was internally like things were going on, but now in the morphodifferentiation it will take form of the tooth that is going to be formed. So this is the morphodifferentiation. And apposition stage. In the final stage, the enamel, dentine, cementum and pulp are deposited in successive layers, resulting in the formation of fully developed tooth. Okay, but what is important in these stages is that they overlap. Okay, so initiation does not overlap. It is just first there's just interaction between dental lamina, oral ectoderm and the ectomesenchyme. But proliferation, okay, so this growing and development of the buds, it will take place in the bud stage as well, cap stage as well, and the early bell stage as well. So the proliferation goes on throughout three stages. In a similar way, the histodifferentiation stage where ameloblasts and odontoblasts are formed, it will start from the cap stage and go on till the advanced bell stage. Similarly, morphodifferentiation. So the shape of the crown has been taking place from the advanced bell stage and enamel matrix formation and root formation. So, sorry, not in the root formation, but the shape of the crown has been determined throughout the advanced bell stage and enamel matrix formation. So, this stage overlaps between the morphodifferentiation overlaps between advanced bell stage and enamel matrix formation. apposition in the final stage. So apposition is the final stage where the whole dentine and cementum and everything is laid down. So this takes place in the enamel matrix formation and in the root formation. In the root formation, the dentine and cementum is also being formed. So this stage is present throughout these two morphological stages. So you have to just understand that which physiological stage is going on during which morphological stage. This is the whole point of this topic. So that this was a bit development of tooth. If you like the video, like, share and subscribe, and for the notes you can check the description. Thank you.